Imagine a battlefield for a second. A single sniper silently eliminates a target, reloads, and takes down the next one. Dozens of enemies totally neutralized by just one relentless agent. What if a drug didn't just block a cancer protein, but actually marked it for execution, walked away, and did it again and again inside your body? Okay, let's dive into this explainer because today we're looking at a truly revolutionary medical paradigm. Section 1. The drug discovery problem, a biological limitation. For decades, medicine has really relied on this classic lock and key model. A drug molecule essentially had to perfectly plug the active pocket of a protein to block it. Basically, one drug blocks one protein. But here's the massive limitation. Roughly 85% of human proteins, including things like transcription factors, completely lack these pockets. So under the old model, they were entirely, frustratingly, undruggable. And that forces a really fundamental shift in how we think. Instead of what's called occupancy-driven pharmacology, plugging the lock and just crossing your fingers that the key stays put, we move to event-driven pharmacology. We don't need a pocket anymore. We literally just grab the protein and throw the entire door right into a shredder. Section 2. What is a PROTAC? Now, what's really interesting about this slide is how it introduces our relentless protagonist. So PROTAC stands for Proteolysis Targeting Chimera, and what it does is utterly fascinating. Instead of just inhibiting a target, it brilliantly hijacks your cell's natural waste disposal system, literally turning your own cellular machinery against the disease itself. Engineered kind of like a 3D dumbbell, it has two grabbing arms connected by this flexible linker. One arm binds tightly to the disease-causing protein. The other arm, well, it grabs an E3 ubiquitin ligase, which is a natural enzyme your cells already use to dispose of old proteins. So the PROTAC acts as a molecular matchmaker, forcing these two strangers into a lethal embrace. Section 3. The lethal mechanism step by step. You have to remember, the PROTAC is a catalyst. It triggers an event without being consumed itself. First, it bridges the target and the E3 ligase into a tight trio, forming a ternary complex. Second, the ligase slaps multiple ubiquitin molecules onto the target, which is essentially a biological kiss of death flag. Third, in a brilliant move, the PROTAC detaches completely unscathed, recycling itself to hunt down another copy, like a hitman that never gets caught. And finally, we get destruction. And this brilliantly illustrates the end of the line. The tagged protein is dragged over to the 26S proteasome, which is basically a cellular wood chipper. It unfolds the protein and shreds it into harmless peptide fragments. Absolutely nothing remains. This right here is event-driven pharmacology. The PROTAC triggers one event, the death warrant, and then moves on. It's catalytic, it's relentless, and it's effective at vanishingly low doses. Section 4. Real-world impact. VP and U. Now, let's shift from theory to clinical reality, because this isn't science fiction. In May 2026, the FDA granted approval for Vepinu. This is a monumental milestone, folks. It's the very first PROTAC therapy approved for human use, specifically developed for advanced breast cancer. Administered as just a daily pill, Vepinu hijacks the cereblon E3 ligase to systematically eliminate estrogen receptors. And those receptors are the key growth signal for these tumors, driving resistance when patients stop responding to standard therapies. Imagine it. In real time, tumor cells literally starve and lose their growth signal as those receptors vanish one by one, shredded by the cell's own proteasome. As Dr. Noah Berkowitz notes, Vepinu addresses a massive unmet need for patients progressing on initial therapies. So the crucial point is this. By actually degrading the target rather than just temporarily blocking it, this molecular hitman simply shreds the underlying mechanism of resistance entirely. Section 5. Erasing disease forever. Vepinu is really just the beginning. The clinical pipelines are swelling right now because PROTACs are cracking open that remaining 85% of the undruggable proteome. We're talking everything from aggregation-prone proteins in neurodegeneration to viral proteins. As we wrap up this explainer, realize that we are witnessing the birth of an entirely new therapeutic modality, one that doesn't merely silence disease at the protein level, but erases it. The question is no longer if PROTACs will reshape medicine, but how far the revolution will reach. Degrade, recycle, cure.